Good morning class. Today's topic is types of ovule and the pollination. So here, for the types of ovule, based on the arrangement of micropyle, chalazal end and the funicle, based on its arrangement, there are different types of ovules. Okay, so the first type of ovules is orthotopous ovule. So here the micropyle, chalazal end and the funicle end. The micropyle, chalazal end and the funicle end all are in the same plane. So that is called as a orthotopous ovule. So this orthotopous ovule, it is also called as a upright ovule. It is also called as a upright ovule. So here the example, it is mainly observed in gymnosperms. It is being observed in the gymnosperm. It is called as a orthotopous ovule where the microspore, chalaza and the micropyle, chalaza and the funicle all lie in the same plane. So next type it is a anatropous ovule. So this is a funicle end. Okay, so the opening is a micropyle and here the opposite of a micropyle, it is a chalazal end, right? So we have already discussed about the anatropous during the structure of a megasporangium, right? So here the anatropous ovule, it is also called as a inverted ovule. It is majorly observed in the almost in the 60% of angiosperm. We can observe the anatropous ovule. So it is being observed in the angiosperms. So here the chalazal end and the micropyle end will lie on the same plane, whereas the funicle will be in the opposite plane. So that is called as a anatropous ovule. So next type of ovule is a hemitropous ovule. So here this is micropylar end, this is chalazal end, and here it is a funicle end. So this is a embryo sac. Here the funicle end, okay, the funicle end and this chalazal and the micropyle end. So here it is the funicle end is it is almost right angle to the chalazal and the micropyle end. So on the right angle is there. Okay, so here the funicle is this and the chalazal and micropyle is this line. So these two are right angle to each other. So this time it is called as a hemitropous ovule. So this hemitropous ovule, it is also called as a transverse. It is also called as a transverse ovule. Okay, it is also called as a transverse ovule. And the next type of ovule is a campylotropous ovule, which is almost similar to the hemitropous ovule, but the only difference here, so we can see it is a funicle end. So this is funicle, it is chalazal, and this is micropylar end. So this micropylar and chalazal end are striped in white, whereas the chalazal and the funicle end, it is quite bent towards the integument. So there is a straight line between the chalazal and the funicle. The funicle was straight, but here the chalazal and the funicle, okay, the stalk of a funicle, it is quite bent. So here we can see the funicle end is quite bent. So this type it is called as a campylotropous ovule. Understood? So here next is a amphitropous ovule. So here this is a funicle stalk and this is chalazal and this is a micropylar end. So the only difference between the campylotropous and amphitropous ovule is that, so here this embryo sac. So this one is what? It is an embryo sac. So we can observe the difference here, this and this. So embryo sac, it's in a straight manner, but whereas in the amphitropous, the embryo sac, it is in the horseshoe. So here, so the difference is that the embryo sac is horseshoe shape. Horseshoe shape, that is called as a amphitropous ovule. So ಇದರಲ್ಲಿ embryo sac straight ಆಗಿದೆ, ಇದರಲ್ಲಿ ಸ್ವಲ್ಪ ಬೆಂಡ್ ಆಗಿರುತ್ತೆ, ಕರ್ವ್ ತಗೊಂಡಿರುತ್ತೆ. ಸೊ ಅದನ್ನ ಏನಂತಾರೆ, it is called as an amphitropous ovule. And next last one is a syncytiotropous ovule. In the syncytiotropous ovule, this one, it is a very long twisted structure. So that is the funicle. So in funicle it takes one complete turn. So we can see here funicle is taking the one complete turn. So this is a funicle stalk, and here this is a chalaza end, and here this is a micropylar end. So this type of ovule is called as a syncytiotropous. So here the funicle it has been, so it is very long, so it is being curved around this main body of a ovule. So this type it is called as a syncytiotropous ovule. So this was the different types of a ovule based on the arrangement of micropyle, chalazal end and the funicle end. And next we should see the process of a pollination, the general introduction of a pollination. So here the pollination means the transfer of a pollen grains from the anther to the stigma. It is called as a pollination. So pollen grains anther ನಲ್ಲಿ ಇರುವಂತದ್ದು ಸ್ಟಿಗ್ಮಾದಲ್ಲಿ ಪಾಸ್ ಆನ್ ಏನಾಗುತ್ತೆ ಆ ಒಂದು process ಏನಂತ ಹೇಳ್ತಾರೆ, it is called as a pollination. In pollination there are two types. One is self pollination and another one is a cross pollination. Self pollination and cross pollination. Okay, so here the cross pollination, it is also called as a xenogamy or allogamy. It is also called as xenogamy or allogamy. Now let's see what is this self pollination. So self pollination means the transfer of pollen grain from the anther to the stigma of a same flower. It is called as a self-pollination. So here, like in the bisexual flower. So for example, in the hibiscus, so it is having both the anther and the ovule in the same flower, right? So here the transfer of a pollen grain from the anther to the stigma of a same flower, it is called as a self-pollination. So here the definition is transfer of pollen grains from stigma to from stamen to stigma of same flower is called as a self-pollination, or the transfer of a stamen pollen grains from the stamen to the stigma of the different flower of the same plant. It is also called as a self-pollination. So here the pollen grains transfer from stamen to stigma of a same flower of a one single plant is also a self-pollination. Or here the transfer of pollen grains from stamen to the stigma of the same plant with the two different flowers, it is also called as a self-pollination. Okay, so next one is a cross-pollination. So here the transfer of pollen grain from the stamen to the stigma of the two different plants, of the flower carrying the two different plants, then it is called as a cross pollination. So here let's see the definition. So the transfer of pollen grains from stamen to stigma of flower of of different plant is called as a cross pollination. Okay, transfer of pollen from stamen to stigma of a flower of the different plants, it is called as a cross pollination or xenogamy. So cross pollination ಏನಾಗುತ್ತೆ, ಮೇಲ್ ಫ್ಲವರ್ ಅಂತ ಬೇರೆ ಪ್ಲಾಂಟ್ ನಲ್ಲಿ ಇರುತ್ತೆ, ಫಿಮೇಲ್ ಫ್ಲವರ್ ಅಂತ ಬೇರೆ ಪ್ಲಾಂಟ್ ನಲ್ಲಿ ಇರುತ್ತೆ. ಸೊ ಆ ಒಂದು ಪ್ಲಾಂಟ್ ನಲ್ಲಿರುವಂತಹ ಸ್ಟೇಮನ್ ನಲ್ಲಿರುವಂತಹ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಏನಾಗುತ್ತೆ, ಇನ್ನೊಂದು ಪ್ಲಾಂಟ್ ನಲ್ಲಿರುವಂತಹ ಸ್ಟಿಗ್ಮಾಗೆ ಥ್ರೂ ದ ಫಾರ್ಮ್ ಆಫ್ ಏಜೆಂಟ್ಸ್ ಇಂದ ಆ ಒಂದು ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಇನ್ನೊಂದು ಪ್ಲಾಂಟ್ ಗೆ ಕ್ಯಾರಿ ಆದಾಗ ಅದಕ್ಕೆ ಆ ಒಂದು ಪ್ರಾಸೆಸ್ ಏನಂತ ಹೇಳ್ತಾರೆ, ಇಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಅ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್. ಸೊ ಹಿಯರ್ it is of same plant, so here it is of different plants. Okay, in the same plants of flower, it might be bisexual where both the stamen and stigma will be present. So that is self-pollination. Okay, so self-pollination will be easy because whatever the male and female reproductive part is there, it is present in the one single plant, right? Whereas in the cross-pollination, the stamens and the stigma, where the male and female flower is present in the two different plants. So male plant anta bere irutte, female plant anta bere irutte. So ಆ ಒಂದು ಮೇಲ್ ಪ್ಲಾಂಟ್ ನಲ್ಲಿ ಇರುವಂತಹ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಫಿಮೇಲ್ ಪ್ಲಾಂಟ್ ಗೆ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಸ್ಟಿಗ್ಮಾದಲ್ಲಿ ಹೋಗಿ ಅಟ್ಯಾಚ್ ಆಗಬೇಕು ಅಂದ್ರೆ ಬೇರೆ ಬೇರೆ ಆದಂತಹ ಒಂದು ಏಜೆಂಟ್ಸ್ ಇರುತ್ತೆ. ದೇರ್ ವಿಲ್ ಬಿ ದ ಡಿಫರೆಂಟ್ ಏಜೆಂಟ್ಸ್. ಸೊ ದ ಏಜೆಂಟ್ಸ್ ಆರ್ to carry out the cross pollination are air, water, insects, birds, animals, bats, ant and the squirrel. So there are different kinds of agents are there. Suppose the pollen grain is carried by the process of air, by the mean of air, then it is called as a anemophily. Okay, suppose if it is carried by the agent of a water, it is called as a hydrophily. If it is carried by the agents of insect, it is entomophily. In birds, it is called as ornithophily, and for the animals, if it is carried by the mean of an animal, then it is called as a zoophily. Suppose if it is carried by the mean of a bat, it is called as a gynapterophily. Suppose now if it is carried by the mean of ants, it is called as a myrmecophily, and if it is carried by the mean of a snail, it is called as a malacophily. So these are the different agents through which the pollen grains is carried from one plant, okay, from one plant to the another plant by the mean of a agent. So that agents, there will be the different agents, and different agents it is having its own terminology. So we should remember this terminology. So this was a general introduction of a pollination. So here in the cross pollination, so here in the cross pollination, there will be the requirement of adaptation. So the particular pollen grains from one plant to the another, it should be carried to the stigma, right? So here the plant will adopt the certain morphological structure, so that whatever the pollen grains is there, it should be carried on properly. Okay, so this cross pollination, so here the cross fertilization takes place, so hence it helps in the evolution. So the pollination method, it is not much properly taking place because it will be carried out by the agents. Whereas in the self-pollination, so here the pollination, okay, so the pollination success will be more in self-pollination, whereas in the cross-pollination, the pollination success will be less because it will be carried in through the mean of a agent. So here the self-pollination, it doesn't help in the evolution because there will be no more cross fertilizations will take place, so it doesn't help in evolution, and it will be not more stable, whereas cross pollination will be more stable. So in the next class we shall study about the adaptation acquired by the plant to take place the cross pollination successfully. Thank you.